Our speaker today is Harrington, who will be telling us about hypergraphs for multi-scale homology sightings in structured data. You can take it away.
Great. Thanks very much. So I'm mostly going to talk about exactly hypergraphs for multi-scale cycles and structured data, which is now on the archive, I think a week or so ago. But I thought I would actually kind of motivate this talk with some joint work with students here in Oxford and colleagues, which is homology of homologous proteins. So I changed the title a little bit because I am going to talk about hypergraphs or multi-scale cycles in structured data, but I really wanted to give a motivation as well. So it's really a pleasure to speak with you and also to share work that's been done with students and other early career researchers. So this first project I mentioned, there were 4 undergraduate students, and then Catherine Benjamin, who's, you can see, I took a picture from her website. She's now doing her PhD with Ulrike Tillman and me. And then the second project, what kind of came about following on from this project, and that's joint with Annette Barbensi, Iris Yoon, Christian Madsen, who is a PhD student in Melbourne, Deborah Aji, and she's was involved in our Algebra in Africa project that we had. So it was very nice. It was kind of culmination. So when Iris visited before she joined us, all of us were here, and so it was that project started at the very beginning of the year. And so yes, I'm very excited to talk with you about that. If you are interested in this and other applied topology work, I would like to advertise first. Doesn't matter what level that you're interested in. We have a mathematics of data book series with a number of mathematicians involved in that. And so we are looking for book submissions if you are more experienced mathematician looking to contribute kind of graduate level textbooks and texts for the community. Please let us know. We are actively seeking out books and proposals. And then the other thing that I want to mention for early career researchers is that our Center for Topological Data Analysis, the funding comes to an end the year after this one. And so we have 3 one-year postdocs. Also, we have 2 Hook research fellowships. These are wonderful research fellowships. I actually came to Oxford nearly 10 years ago on a Hook research fellowship. Therefore, kind of senior postdocs, and they're independent 3-year research positions with a little bit of teaching. So you get involved in the Oxford system, which is really special. But it's an open call across all of applied mathematics, and so I please encourage anyone that you know to apply. And the final thing is that we're running WinComp Top 3 at UPFL, and so if you haven't seen the link, I think we've sent it through a number of, please let us know and apply. So we'll be doing that this summer. Okay, so I feel like I don't really need to give an introduction, so I basically won't, but this talk is kind of extending the persistent homology pipeline, going from a point cloud to a filtered simplicial complex, and then computing topological features of interest. Here we're looking at H1, and that will be the focus of this talk. And basically looking at ways to really study these topological features individually, and then also the structure of these kind of local features all together. So rather than in other work that we've done where it's kind of this maybe more standard that you would vectorize these your persistence diagram and put it into some type of machine learning or some other classification or various tasks that you're interested in, and taking the whole summary, right? The whole persistence diagram, here we're looking at trying to interpret more specific features. So looking at cycle representatives and generators. And so that's what I'm going to look at a number of examples on how we've kind of been thinking about that in the last year or so. So the first example that I'd like to motivate is where we're looking up I guess the different examples I talk about today all fall into this kind of class of these open curves. They can be entangled, and we want to characterize the topology and the geometry, and in some cases knotted curves in 3 space. And so for looking at, for example, proteins, which is the first problem that we look at, there are many maybe standard approaches for studying these. And so these geometric features, for example, looking at the radius of gyration. So this, you can think of this as kind of looking at the convex hull, but the biophysics and biopolymers look at what they call the radius of gyration. So we want to see like the volume of some specific feature, looking at curvature, an average crossing number when you're looking at some of these problems. And then the topological features, if you have some open, what's called an open knot, you can use knot closure methods or projections looking at knotoids, and then other subchain analysis, which is kind of standard in biopolymer methods. There are many, I mean, this is very well developed and provides a lot of helpful quantification and descriptions of curves such as proteins and other systems. But the challenges are that these are computationally expensive, and importantly, these often require really precise input data. So we can think of, as written on the slide, that these spatial curves, which is what I'm going to call them throughout the talk, these are piecewise linear, one-dimensional manifolds in 3 space. You can think of, well, I guess I'll give you a couple examples, but that's the setting that we're going to look at. So it's very special. From kind of maybe a shape statistics, some people are interested kind of in the space of all of these different shapes. We're really interested in describing features in the space that they live in, right? So rather than trying to quotient or something, we're really studying these piecewise linear curves. The shape, the 3D shape, for example, of proteins really matters. So, right, so why do we care? We can think about these biopolymers that can be modeled as spatial curves. So these are really ordered, the vertices are ordered. And DNA molecules, these could be from short viral DNA to long folded, what are called chromatin, and that's often noisy genetic data that more and more, I mean, there's been other topological data analysis approaches to study that actually, and proteins. So these, we can look at these chains of amino acids, and they form, as we know, right, with protein folding and prediction, they create very complex 3D shapes. And there's this interplay that is really important between the shape of protein, or, you know, in DNA topology, the shape of the DNA, but also what's the function. And we know, for example, that that there's this problem of knots in proteins. Because there's a knot, and the folding of the protein is very, very complicated, actually. So you have this conundrum in knotted proteins, and that's one of the questions that we looked at, which is, so proteins can be modeled as these piecewise linear spatial curves, and we can look at the backbone, these carbon atom backbone. These are think of these vertices as the carbon atoms in our data. And they do sometimes fold to form open knots. And okay, we're not going to answer how these knotted proteins fold today, but we know that if they are knotted, the ones that are knotted are more stable in the sense of, like, thermodynamically, it's more difficult for, or maybe less likely for, that protein to degrade, right? So to deteriorate. if there's a knot in there. And so, so in some sense, they're more, so these are open questions, right? They are, proteins that have knots are more stable in some sense. And so the question is, does the geometry influence the folding and the stability? I think this is more and more traction is getting that this is a positive, that it does, the geometry does influence these, and the knotting does affect it. And so one class of proteins that we study are these trefoil knotted proteins. So trefoil knots are the simplest non-trivial knot, and we can kind of distinguish these two types of knotted proteins based on what are called deeply knotted or shallow knotted. So if you look at the top knot here, you can see that the tail is very long and the knot core is kind of deeply inside from the tail, whereas the bottom kind of open knot here, the bottom one, the knot core is much larger and the tail is much smaller. So that is a shallow knotted protein. And one question, so we had an four undergraduate students look at the fraught database and take in these vertices as carbon atoms. So it was very standard, learn persistent homology, a bit about the theory, and then some computations, take in this data and see if you can compute persistent homology in the first instance of these knotted proteins. And so how did they do it? It was really going from your knotted protein, you have these this carbon backbone for the vertices, and then we just use the Vittori strips, right? So it's an undergraduate project. We meet with them a total of 6 hours over 2 terms. And then they computed persistent homology with the Vittori rips or something in the first instance. And then we asked, well, can we compare different proteins? So we were able to obtain, they computed the persistence diagrams for all of these different knotted proteins to get what we call the fingerprint of that data. And then of course, there's different families of distances on persistence diagrams. So one of them that they looked at 2, and the first was a Wasserstein distance, where we find a pairing between the points in the diagram that minimizes the pairwise distance. And the other distance that we looked at were persistence landscapes, which I think everyone probably also knows, but just a reminder, this is a vectorization method that was proposed by Peter Bubenik, and Pavel Dlatko developed, I think, some of the first implementations of the code. And this is a piecewise linear PL twice. Okay, persistence landscape, right? It's a sequence of these functions where now we can take averages. So we can build these tent functions and then compute different landscapes. I guess I should have written lambda 1, lambda 2, but we have these sequence of functions, and we can compute a distance between landscapes. And so that's what we did. Well, that's what the students did. They looked at these persistent homology of these trefoil knotted proteins. And so here it's known. So if you're looking at the left, right, we have the deeply knotted proteins at the top and the shallow knotted proteins at the bottom. And that's according to depth along the x-axis, and then length of the protein along the y. And so what's called sequence homology representative sounds very topological. This is actually what biologists call very similar protein sequences. They call it sequence homology when the sequence is very similar. Here, so sequence homology representative, this is one example of a protein around a class of proteins that are known to be very similar. So that, I hope that if anybody has any questions, please, but this is not this is the biological names, but this is not right? That's not topology. So what we're showing is what's already known, that we can look at length and depth. So we have some ground truth for what proteins are similar to each other. Okay, and so this was just the biological data that we have from the not fraught database, and the question and PDB. And so the question is that, can persistent homology detect different knot depths? And that was our first question, right? Because there's a ground truth, we know what this is, and then we wanted to see, can persistence do that? So we can partition the proteins, exactly what I showed you before, into these structural homology classes, and we then label them. They're either deeply knotted, shallow knotted, or neither. And so this is other work that's been done. So we've labeled them according to what's known. And then we created a pipeline. So we took the point. So from the proteins, we have a point cloud, compute persistent homology to get a persistence diagram. And then from the persistence diagram, we can compute topological statistics. So we compute landscapes and average landscapes for these sequence homology classes of these structural homology classes of proteins. And then we can go into that. And what we found is that between knotted and unknotted, so what we can also do is look actually at one point in your persistence diagram and using Irene computing a generator. And so we could get that a basis element, and that's what you can see here in red is one of them that we looked at. So I'll explain a little bit more. So we can both look at the persistence landscapes, but then also some of the local geometry in the protein. So the first thing we looked at was with these two different distances, computing persistent homology of many different proteins. And we looked at the distances, and what we found is that a global clustering, we could cluster both by the sequence homology representative. So you can see that similar proteins all are similar, and we need to use both the Wasserstein and the landscape distance to jointly cluster these trephile proteins. And then and we can do that with the structural class, but also with the knot depth. So these are either shallow, deep, or neither knotted. And you can see that it does a pretty good job. And I was impressed. I mean, I was surprised in some way because I don't know, we don't know if we expect it, but then we also can, from this, then say, okay, all of the proteins in one of the sequence homology classes, we can compute an average. So we can average the landscapes to get a summary of geometric features of one of these classes, and then we can confirm statistically that these have different averages. Now, what I think is very cool is that based on this, we then ask questions about the knotted and unknotted proteins. So we wanted to localize, what we did is we looked at the average knotted and the average unknotted landscapes. And just by looking at them, we could see that the second landscape showed this clear difference. You can see this kind of blip at the bottom right of the knotted protein landscape. And so then we used Irene to actually localize what was happening with that specific generator. So we looked at this basis element, we looked at the vertices that were involved, and it precisely localized this crossing change in the knotted versus unknotted protein, that when these have a very similar sequence. So these are homologous proteins, so their sequences are almost identical, but they differ because we know that the topology is different. We know one is knotted and one is unknotted, for example. And so we could actually distinguish these knotted, what are called AOT cases, and unknotted OAT cases by this crossing change using these persistent homology generators. So just from the landscape, which is pretty cool, right? I think it was because, okay, so this is stuff that I said we only had 6 hours with undergraduates. This continued. This was, so Catherine Benjamin, who I mentioned, did a lot more work after the project ended and during part of her PhD to actually, well, yeah, so for a good start of the PhD, she started doing this and this is the result, which is pretty exciting. The next question we asked, well, is this robust to noise if we do that? And so, I mean, we have the data, right? We have these proteins, the non-proteins, and we can add noise to the data. And so so Catherine added more and more noise to the data and found that the persistent homology was pretty robust, which I guess is what we'd expect because we have results for stability theorems for persistent homology, but we don't have them necessarily for certainly we know that these are the cycle representatives and the generators are not they're not unique. We don't have any stability results that I know of. Maybe somebody here will tell me actually, but so we added a lot of noise and just asked, well, can we still distinguish them? And we could still cluster not as well, but we could still cluster even with some noise. So the second part of my talk is looking at more noisy data, right? Because protein data is very, it is precise. It's useful for standard kind of not theoretic approaches because it is very perfect. But when you're adding a lot of noise, this is one of the strengths of topological data analysis. So we started looking at more noisy data, noisy spatial curves. And so based on this, that's kind of this like the second part of my talk. Are there any questions? Is that okay just to ask now before I move to the next? Okay. So then the second part of my talk is looking more generally at these persistent homology of spatial curves. So again, we have these ordered points. Oh, is there a question?
Yeah.
Okay, we have these ordered points. Yes.
So my question is, what type of noise you added to the proteins? We know that the proteins are not sort of stable; they are fluctuating by naturally. But I'm just interested, like, what type of noise you added, and you still have the same sort of topological properties.
So I think that we added Gaussian noise to each point, and we just increased the amount. Yeah, so I mean, when you look at I can't remember now where, I mean, when you're adding so much noise that the curves we don't show how the proteins look, but they look like a mess at some point. But it's just Gaussian noise to each of the vertices, the location of the vertices.
Yeah, great, thank you.
Okay, so then the second part is looking at these spatial curves, these order points in 3 space. So we have a curve, and what I mentioned before is that we looked at one of the points in the persistence diagram, right, given by differences in the landscape, and then from Irene computed, because Irene's very fast at computing these generators. So we could compute this basis element and then highlight which vertices. And one question we asked is why are we doing it only for one point in the persistence diagram? What happens if we do it for all of them? And so we've proposed an extension for analyzing some of the higher order interactions at the level of generators. So here's we just have highlighted these 3 different generators corresponding to the same color in the persistence diagram that we computed from Irene. So this is the standard pipeline. We have point cloud and persistent homology. We've chosen some filtration, and then you choose some way of computing your generator. And I think everybody knows what generators are, but right? So we would, these are not unique. And we know we use Irene where the generators are not canonical, they're not optimal, and other software may give different generators. So Greg has given me a slide to talk about Irene, which computes a basis for the whole chain complex before returning a persistence diagram. And these are notes. So each point in the persistence diagram corresponds to an element of the basis. And when the user asks for a generator, Irene returns that basis element. So we call these matroid generators in this work. And the matroids provide a proof of correctness for Irene's main algorithm. So every persistent homology calculation can be expressed as a calculation of a min weight basis of a matroid. So in dimension 1, that can be expressed as a minimum weight spanning tree. So the matroid theory provides the theoretical guarantees that all the batching they do is correct, and then uses heuristics to minimize. But as I said, right, there are some problems or some challenges that, I mean, I think Irene's amazing, but there are some challenges, which is that we don't have the uniqueness, like I said, it's not canonical, it's not optimal, and it doesn't include the fact that the data we're looking at is very special. It's these, we have these ordered vertices, so we have this curve, and because of that, we then, Iris actually calculated minimal generators. So we take our curve, we can compute the persistence diagram, and then she uses there's some work by Lee et al. Where they have these minimal cycle representatives. So you can do length minimization with rational coefficients, and the experiments show that most minimal generators computed with rational coefficients have coefficients in 0, 1, or -1, so we can interpret these coefficients over the binary field, and so there's edge cases, but actually for our problems we didn't encounter any of them. And then what Irene did with what Iris did was compute these length minimizing generators. So the idea is that you can see, because this is a curve, we want to minimize the number of jumps so that we are really preferring the homologous cycles that minimize the jumps between non-consecutive vertices, because these vertices are ordered, they're coming from curves like proteins or other curves that we'll talk about. Okay, so I was very surprised. We computed both on all of the data with minimal generators and matroid generators. And when you visualize them, they look actually remarkably similar, which I think might be a result of the data set that we're looking at is very special. The data sets that we look at have this special structure. But by eye, if we take our, if we take our curve and compute persistent homology and then compute minimal and matroid generators, and you look at them by eye, I mean, they're a little different, but they are, I mean, the first, right, we see that the first cycle does pull out the same, approximately the same vertices. So these cycle representatives appear to be, yeah, and even though the minimal generators, the computations are more expensive, the matroid generators with Irene are very fast. We ran everything with both, but everything we present is with the matroid generators. I'll show you the comparison in just a minute. Okay, so so then how do we compare them? How do we analyze this, and what do we get out from it, right? So Inesi had this brilliant idea of what about thinking about these. Each of the points in your persistence diagram corresponds to, we can localize each of these generators with a weight, right? Because there's a persistence that's attached to each of these generators. Why don't we think of this as each of the vertices for the generator that we've computed, why don't we think of that as an edge in a hypergraph? So just to recall, a hyper edge is just a subset of your vertices in your data, and we can attach a weight to that, and so that's a weighted hypergraph. And then in this case, we call this the persistent homology hypergraph. So we do this for all of the points in our persistence diagram, and that encodes all the kind of local as well as global topological information. So here we're not thinking of analyzing the persistence diagram. We want to analyze the persistent homology hypergraph. Okay, so we're really looking at, if you want to think back, right, we're really looking at the top row here in the generator, right? We're looking at each of those vertices are assigned, so that loop, that cycle in the data, that loop, it's assigned a hyper edge, weighted according to the persistence, and again and again and again, and we do that for all of the points in our persistence diagram. So we're looking at the connectivity in some way of the data, but we're keeping this higher order, the local and global topological information. It's not just these curves and the sequence of the curve, and it's not where they are only in space. We want to include both of those.
And Heather, just a quick clarification question from the chat. So these generators, are they from the Viator-Scripps construction or from a different construction?
Yes, but we don't restrict, right? So we use Viator-Scripps, but we don't have any theoretical statements that prove, right? I mean, we're trying to make it as general as possible. So, and that's something that actually there's a number of things that we're interested in looking at, and maybe there's some nice, this is one particular structure of data. We want to look at more general. Also looking at other filtrations would be very nice, I think. Exactly. But yeah, so we did this with Viator-Scripps. And so you choose whatever filtration, and then you can choose whatever generator way you want to compute your generators. Since I've done this, I've learned that there are many other software that can compute generators, and it would be interesting to compare all of these. We haven't. We did these two. Okay, and then once we have this persistent homology hypergraph that encodes, right, the generators and their persistence, what do we do? And so this is the pipeline we're proposing, which is analyzing our persistent homology hypergraph, and we do that with what we call persistent homology centrality and persistent homology communities. So I'm going to tell you a little bit more about these in a minute, like exactly what they're computed, but I can at least give you a rough interpretation of what persistent homology centrality is. So we've gone into network theory, network science, and there's been an explosion of methods to analyze hypergraphs in the last couple of years. We wrote this recent review article, What are Higher Order Networks? And many of the more recent papers, we tried to include a section in hypergraphs. So for those interested, so we use centrality as a measure of importance. So what nodes are most important in this setting, which nodes. So we've assigned each vertex a value based on how important, how many times that vertex appears in a generator, weighted with some weighting. And so there's a method out of the box method we use, but that gives you an importance of which nodes are most important. I think that maybe, okay, so that's pH centrality, and then pH communities is partitioning your, so often you have some point cloud data all the way on the left, right? And you want to cluster, maybe build some network and then cluster it, build some communities. And these are a disjoint, it's a partition of your vertices, and we partition based on the persistent homology hypergraph. So the edges, the hyper edges. And so so we've computed communities from our pH hypergraph. these, that encodes, right, the higher order interactions from the generators and the persistence. So that's our pipeline we're proposing. I think there's lots of interesting things. That's why I want to talk about that today, right? Is I think there's a lot of interesting problems and questions that can be further analyzed here. I think this is just a kind of first proposal. You could probably do something completely different than what our choice of H was, how we analyze our pH hypergraph, but these were the two that we chose. Okay. And then, so it appears that we looked at these curves of different lengths, length 100 vertices, 200 vertices, 300 and 400. And we looked at the correlation between minimal generators and matroid generators. So we wanted to quantify, whether these correlate if we change them, and we find that that they correlate pretty well. And so that's why we chose to, even though we computed everything for all the data sets with both, which took a little bit more time, and we in our archive paper, the matroid generators are what we have in the main paper, just from Irene. But you can see they correlate pretty well. So then we wanted to further analyze this hyperTDA pipeline, and the first thing we do is look at what's called node centrality. So this is the ranking of the vertices using this hyper edge membership and the persistence, the weight. And the importance of a vertex depends on the importance of its connections, and that's the idea. So there's something called eigenvector node centrality, and so that's what we used, which I've included the reference here. You can see it's 2021, so it's a very recent centrality. It's a spectral centrality measure, and it looks at high values, so large values of centrality to nodes that are part of at least one other important hyper edge. So you can find these nodes that are involved in many different generators. So these are kind of the key or the most important nodes in your data set. And so you can pretty easily see, so if yellow is highest centrality, then you can quickly see on the left, these are just hand-drawn curves. On the left, you can see that the yellow is kind of in the center of this flower. And then on the right, where you have like this roller coaster, it's exactly where you would see that a loop could form with Vittor's rips. So you have the right in the center, those have high node centrality. And then we want to see, well, how does this correspond with real other problems and other data sets? So we looked at movement of different species. And so this is a nematode, it's a worm, and this is data that was we used from, so this is a collaboration with Michael Stumpf and AnDi, who's in Michael Stumpf's group, and Christian. And so in a previous paper by the Stumpf group, they analyzed the movement of these worms. And so we had this data, and we computed the hyperTDA pipeline and node centrality. And what we found is that the centrality actually could distinguish the different behaviors, which are pretty well known, that worms go through different areas of behavior, where it's in looping is high centrality, it's in the bottom right. And then they go through low centrality, which is kind of this search, and the relocation, again, with slightly higher centrality. So, oh, sorry, I switched the search. But anyways, you can very clearly see that centrality kind of distinguishes different movements of the worm. And we looked at different tracks and found this. And all of them are in the archive preprint. Okay, so that's kind of cool. And then we said, okay, well, what about the persistent homology communities? What do we learn by analyzing those? So community detection, right? It's a partition of the vertices, and the way that community detection works. So we use what's called the Louvain algorithm, but you're wanting to find densely connected groups of vertices, and densely connected in the sense of these hyper edges. So we and so by each of these partitions, these communities, they represent, they're often thought of representing functional modules. by these persistence generators. So we do flatten it to a graph for computational purposes, and we include all the edges, but within each of those generators. So then that does bias it a little bit, but in terms of being able to compute community detection, we flatten the pH hypergraph to a graph to compute these communities, but it does keep all of the, it does maintain the, it's not on the initial point data, point cloud data or a network built on that. It is on the generator. So, so we then said, well, what can we learn from this and what can we see? And so we looked at this hand-drawn spatial curve, and you can very clearly see that each of the communities, which are given by a different color, can identify functional modules in this flower. And so we get this density matrix. Whereas if we looked only on the point cloud data, for example, the structure would be difficult to detect alone. So I mentioned that this works really well with noisy data, and in current, like a current in progress manuscript that we're working on with some collaborators with the Michael Stumpf group and his collaborators is looking at this problem of folded chromatins. This is looking at this very noisy genetic data where you're looking at the 3D organization of the genome, and you can see the ground truth and the pairwise distances. And then there are standard methods, which really are looking at what's close in the curve in some way. And we want to have something that can encode this higher order interaction. So we do this hyperTDA, and our TDA analysis actually picks up these persistent homology communities that are larger, and these are more like functional modules that the biologists that we're working with are super excited about. That the pairwise distance, right, there isn't too much there. The standard domain, the standard analysis that's done is really only local, and here we're bringing kind of both this local and global connectivity with this hyperTDA pipeline. So that's kind of in progress work. Then the next question you have, well, that we had is hyperTDA, are we just picking up things we already know about and can compute? So we looked at these 4 different lengths of randomly generated curves that they're self-avoiding walks. And we computed centrality and persistent homology communities. And then we looked at the correlation of the centrality, which is the top row, with other geometric measures, torsion, curvature, and density. And these are computed with pi nautic. And now I can't remember our definition of density. I can look it up. If somebody asks me a question, I can look it up. But you can see, so density seems to correlate well. I think that's like looking at every, no, I'm gonna get it wrong, but I can look it up if somebody has a question. And then, but we can see that torsion and curvature don't correlate at all with the centrality. And then we looked at the community, persistent homology community matrices and the pairwise distance matrices, and there's no correlation. Again, it's really bad. So we wanted to try to understand what we are picking up in these pH communities. So the next thing we looked at is what we call the spatial volume. And so this is looking at the biologists call it the radius of gyration. The way I think about this is looking at the convex hull of the vertices in this community based on their 3D-dimensional location. So we can compute spatial volume, we can compute the number of vertices in each of these communities, which we call the pH community size. We can compute geodesic distance, which is from looking at the vertices that are assigned to each community because this is an ordered pair, ordered set of vertices, what's the first and what's the last. And so you can see that, for example, the yellow community has a geodesic distance 9 because all of the vertices are within the same portion of the curve. Whereas, for example, the red community, if you look at the left picture, you can really see that it's kind of at the beginning and the end of this curve. And so it would have geodesic distance 37. So we use these measures of geodesic distance, persistent homology community size, and the spatial volume to compute, and geodesic intersection. Sorry, this is, we're actually looking at a statistical test for looking at the intersection that we want to quantify the discrepancy between the spatial and the geodesic matrix metrics. And so we can do this with a statistical test to get a p-value. So we would see, I hope I have a slide that gives you some indication, but that's another measure of the pH community, is a difference between kind of the local and the spatial and the geodesic distances on these curves. And then with these measures, we then looked at network. In network science, there's this notion of assortativity, which is that nodes that are more similar to each other are assigned to the same community. And so you can see the very left, we call it low ambient and low intrinsic assortativity. And the way that we're computing this is looking at ratios between the size and volume of the communities. And these are so the community size is the numerator, and the denominator is the volume, the spatial volume. And then the intrinsic assortativity is the average ratio between the community size and the geodesic size of the communities. And so we get some measure of high and low assortativity, how similar, again, vertices are that are assigned to the same community. And we try to, so we can kind of interpret a little bit, and then we want to apply it to a couple different spatial curves. So one of the applications that we looked at is anomalous diffusion, transport of diffusing particles. And these are ubiquitous in nature. They're used a lot in finance as well. So you can think about movement, and there are 5 different models that can generate this type of these types of curves, these trajectories. And this was a challenge in 2020, is can we distinguish different models? And I've written what these models are because I'm not an expert. If anybody is very interested, I can go into it later, but I think that just to give you a feel that these, you can see the trajectories do look very different. The top trajectory, we have the different communities, and the bottom is the centrality. And so we can then use the measures I mentioned to look at to compare them. And so we then look at the communities and we find model specific differences between for example, the geodesic distance. And then we use this to look at ambient and intrinsic assortativity. And what we find is that the different models have different, that we can kind of interpret it. And this is a little bit outside of what I'm comfortable talking about, because some of my co-authors were the ones really involved in this further analysis. But we then took the information from these persistent homology communities, as well as the weight of, so we have the persistence, and we put that into a CNN architecture to ask, can we distinguish different models? And we can distinguish 3 of the models with pretty high confidence. So that was a very nice application to be able to interpret, but then also link hyperTDA with some machine learning to, okay, so we didn't rank top, but at least competitive in the top few other competitors in this AnDi challenge. So this seems like a nice approach to distinguish different trajectories. And then I just have one more application, which is looking at immune cell tracks. These were neutrophil cells, zebrafish embryo. And so what they did was in some of them, some of the zebrafish, they cut the zebrafish, so there's a wound, and then it's known that these are these zebrafish, the immune cells will move towards the wound. So there's some bias in some way, I guess, of the zebrafish tracks. And then what you can see is that the zebrafish tracks that have this bias, as well as simulated data that has bias and unbiased tracks, that the plot on the left, you can see that the bias and the zebrafish are almost indistinguishable in terms of the hyperTDA analysis of the geodesic distance, the community size, the volume of these persistent homology communities, and their geodesic intersection. So we looked at these different properties. So that was really encouraging that we thought, so that the biological assumption is that these zebrafish tracks have a persistent biased random walk, and they use persistent not in a persistent homology. It's like everywhere in biology, some of the same words keep popping up that have many meanings. Okay, I am nearly out of time. I'm sorry, I didn't mean to talk on forever, but let me wrap up, which is that we looked at the 3D structure of spatial curves, that we looked at many different applications, 3D genome organization, animal movement, cell movement, that this seems to be kind of an interesting approach that we're going to explore further, but it seems that we can compare different, that kind of unifies and compares different metrics, along the geodesic versus in the Euclidean space of these curves. We get some higher order interactions, and we can distinguish some features that are indistinguishable with a persistent homology from the persistent diagram alone. So, and on top of that, it seems pretty robust to noise, to choice of generators. And I think I'm going to skip one of the extensions, but I'm going to end on a slide to thank many of the people, many of my collaborators here that I mentioned, and just to remind you that we are advertising and recruiting, and would love for you to spread the word and to apply. So, thanks.
Thank you very much. So before we go to the questions, I would like to ask everyone to unmute themselves, and so that we can clap together. Thank you. Okay, so I see there is already one question in the chat that I will read out. Have you looked at any applications where your TDA analysis failed?
That's a question I always ask of other people. So I really wanted to close the loop in some way, right? So I started with proteins, right? And then we looked at the animal tracks and all these things with hyperTDA, but I didn't go back to the proteins. And you might ask why. And the point is that we couldn't go back to the proteins because the proteins have different lengths. And all of our analysis in some way for the hyperTDA required that the size, the length was the same. Otherwise, it's very difficult to compare. You can imagine, how do you compare systems with wildly different numbers of vertices? So what we did in other systems was subsample, which was completely fine, right? We just took snippets of the AnDi challenge, but you can't do that with a knotted protein. You need all of that, right? So that's something that didn't, so Analyse is now in Melbourne, and she is a student that's looking at extending this to handle that, those scenarios, but it wasn't something we could easily solve here. So yes, that's something, thanks.
Great, thank you. Does anyone else have a question? You can just unmute yourself. Yeah.
Sorry. Hi, thank you so much for the talk.
Hi.
And I have a question because you said many times that this pipeline is robust and like the choice of generators and everything. But like, is that something that you're observing in the many simulations and all the applications that you're doing, or did you actually manage to prove that everything is robust to noise and all of those?
No, we didn't prove it. No, we didn't prove it. I think that would be great, but I don't know how to start even actually with some of this So
Probably it's
think
very
that's-
difficult, yeah.
but I mean, yeah, I think that would be great.
Yeah,
it was just what we saw in all of our experiments, right? We tried to do as much as we could with the correlation and be as as kind of as thorough as we could with the even the synthetic data.
Okay, thank you so much. I mean, you like looking at it, it looks really nice.
Oh, thanks.
I have a question. Heather, thanks for the really nice talk. So I had a question on your slide about pH communities. I mean, maybe you don't need to share the slide again, but you had these graphs where you had sort of red spirals and blue nodes and yellow nodes. And on that slide, I lost track of what the nodes were. Were the nodes cycles, or were those just example graphs?
Okay, I can share the screen. Hold on, let me just see if I'm. But I can just tell you, okay, so we had each of the vertices were assigned some community.
Oh, okay,
Right.
I see. So
And so
you,
that was just a
you
color.
had the hypergraph, and then you did the community detection.
Exactly. So we have our pH hypergraph. Everything was always analyzed on the pH hypergraph. And then we just used two possible measures, which we called pH centrality, which was this node eigenvector centrality, or the pH community detection, which partitions the vertices in our hypergraph according to the pH hypergraph of ones that were most densely connected. So basically all of the vertices that would be assigned to one community were more densely connected in amongst generators, I guess, is the idea. And so some of those communities we would find are not, and we did actually, right? Some of the communities you see are actually like loops rather than just dense clusters, because it's not spatial dense clusters, it's organization in the biological, in the curve. Yeah.
Very cool. Thanks. I see. So it's a community detection on these persistent homology hypergraphs.
Hypergraphs. Yes. Yeah. So it's a very, like, I cannot tell you how long I spent trying to get my head around it. So I'd really be open if other people have ways to better explain it. I mean, we really, right, because it's so much analysis, it feels like you're almost like in an onion, and you're like, how do you, yeah, I hope you're not still, you probably are recording still this, the question.
Well, we can cut it out if you prefer, but yeah.
That's fine.
Does anyone else have a question?
Yeah, do you mind if I ask a follow-up on that?
Great, yeah.
So, sorry, and I don't have my camera. Well, here, I can turn my camera on so you can see what's going on with my life. Did you see any comparison between, or did you do any comparison between the clustering in the pH hypergraph versus just naive spatial clustering of the points, or
Yes.
something more naive?
Yes.
And maybe
Yeah,
even show that.
absolutely. Absolutely. Yeah, absolutely. Let me just see if I can share this again. Let me see. Can you see this picture?
Mm-hmm.
Okay, so we have, exactly. So we have points in curve points, the three-dimensional points. And if you do pairwise distance,
then you try clustering that matrix and you get just you'd probably just get like that one big cluster, or like maybe the tail on the left and the tail on the right.
Yeah, exactly. And then whereas with standard, exactly, you don't get very much insight at all, because that's what many people have done looking at the chromatin problems. And then there are other standard methods, which is just really looking at what points are close along the curve. And that's where they have these what are called standard domains. And ours kind of can, this hyperTDA seems to be able to use kind of both this local and global connectivity to give these functional modules that the biologists seem to be very excited about. Yeah.
And just a final question, how big are these? How many points are these? Like is this like hundreds of vertices or thousands, or?
Yeah, the chromatin is it's bigger, but we use, I think, snippets so that we can compare.
Okay,
Yeah,
thanks.
I could look up exactly that number for you. I don't have it at hand.
Okay, nice. We had already many great questions. Maybe we end the recording here, and then you can still ask questions afterwards if you want. So thanks again. Very nice talk.
Thanks for the invitation. It's great. Thanks.